of Cultural Science Universitas Hasanuddin in collaboration with Association of Indonesian Archaeology Sulampapua. Thank you so much for taking a part in this webinar. We are pleased to have you joining us and to take a part in today's session. Well, in order to be considerate and respectful to everyone attending our presentation today, all participants are mute. You will have time during the session to ask questions, but you might submit questions at any time during the session. Submit a question to our panelists. Please enter your question in the Q&A text box. So if you experience any technical difficulties, please send a chat message in the chat box. Likewise, please turn on your video screen and change your webinar's name into your institution. Okay, today we would like to talk about the Archaeobotany, food and plants, food identification, and environmental reconstruction through archaeobotanical studies. The two speaker will give the presentation today. The first one is Basira Mir Mahamad. She is a PhD candidate student in Archaeology Department in Max Planck Institute for the Science Human History, Germany. And the second one is Dr. Wawan Sujarwo. He is a researcher in Ethnobiology Research Group, Research Center for Biology, Indonesian Institute of Science, LIPI, Jakarta. Both speakers will give presentation during half hour, and for each questions also half hour. The attendance list will be shared at the end of the presentation, so make sure you will follow the whole webinar until the end. Okay, due to our time is limited to the first speakers. Basira Mir Mahamad, time is yours.
Hello everyone, can you hear me? Hello. Yes. Because I'm a bit worrying about my internet connection. Sometimes it's written that I have a bad internet connection. Also today is Saturday, not too many people in the institute, but still. Okay, I will start. Thank you for the introduction, and I'm grateful that you asked me to give a brief introduction to the archaeobotany as a field and what we are doing, how we are sampling, and I would be happy to share with all of you. Okay, my presentation. I don't know if you, can you see the presentation?
Yes, we can see.
Okay, I should start. First of all, the title of the presentation is just simple, that is the archaeology botany axis, because I would like to show how archaeology is related to botany and how they together contribute to the archaeological science. And let's start from the beginning. I would like just to tell you briefly what is archaeobotany is and about terminology. This field of study first termed ethnobotany. Today is termed either archaeobotany or paleoethnobotany. Ethnobotany is more common term in Europe, whereas paleoethnobotany is a term used in North America. Comparing definitions of these two terms, both of them are similar in as much as both are study of archaeologically derived plant remains. It's expected, it expanded tremendously as a field at the end of, in the second half of 12th century, 20th century. But to be honest, from my own point of view, I think that it became more popular only last few decades. And also the question which people ask, are you an archaeologist or are you a botanist? And you can be an archaeologist but study archaeobotany, or you can be a specialist in botany but you can do archaeobotany, and it's not so important who you are in order to study archaeobotany. And why do we need archaeobotany? First of all, from my research, I'm interested in agriculture or arboreal culture. And because it's played the main economic role in the early civilization and its development became a jerk of human progress. Therefore, materials related to agriculture contribute to the reconstruction of a clear and accurate picture of the past. And it's a first reason why we need archaeobotany, but there are many different reasons like why we have to study archaeobotany. First of all, it allows us to trace climate changes or make environmental reconstruction, or even like reconstruct ancient vegetation, paleodiet reconstruction, and to understand human-plant interrelationship, because it's also important how human influence, like how people influence to the plant development. We know that before plants, when they were wild, in a wild form, they have a bit different shape. When they became like domesticated, cultivated, the shape of plants was changed also. It's very important. Okay. First, and the second part is we have two like kind of types of plants. First is macroremains and microremains. I'm just working with macroremains. Maybe in the future I will study microremains, but for now just macroremains. But I will give you just a brief introduction about microremains, although I'm not a specialist in this field. And as you can see, it's pollens, phytoliths, and diatoms, starch grains. All of them are considered as a microremains, but I will just discuss two of them, pollen and phytoliths, because I studied about those two at university, in Sapienza University. And first of all, phytoliths, what it is, it's, in other words, it's a silica skeleton from plant tissue. And they survive after a plant has died. And their analysis can provide valuable information about the use of space within a structure of or within some archaeological site. As a pollen grain are produced in a varying amounts, shape, and sizes, which actually are helpful to identify not to the species level, but family, maybe, yes, to the family level. And its palynology can be useful tool in reconstructing the vegetation cover of landscape in the past. But it's very important because most of you are archaeologists, and maybe in the future you would like to introduce archaeobotany to your, to use as one of the methods in your studies. And it's important to remember that when you're taking samples, you have to be very careful. You have to avoid contamination, because also be careful with the pollution from tools and air. Also, it's better. I think it's better if palynologist or an archaeobotanist will take samples. It's as I think. For example, for both of them, we have different sampling strategies. It could be vertical, horizontal samplings. It depends again from your research question. And the amount of each sample should be, when I was at university, my professor told us that it should be around 50 grams sediment samples in a stratigraphic order. But I read many different papers, and it's written that it's still difficult to say about the amount. It could be 30 milliliters, or it could be 60 milliliters. But to be honest, I think if you take more, it will be better for you. And as I already said, pollens are really good for paleoclimate variations, if you would like to know about paleoclimate variations, and natural and human-induced vegetation changes. but phytoliths are also good for grass identification. And next, macroremains. Carpology is my favorite part because it's what I'm doing. Because I would like to highlight that macroremains, we have carpology and anthrocology, wood analysis. I will tell about it later. But the samples, first of all, could be taken not only by an archaeobotanist, but also by an archaeologist. Macroremains, are plant remains that are large enough to be seen with the naked eye, and that can usually identified with a low power microscope. And as you can see on my presentation, I mentioned only about seeds, fruits, and nuts, but it's not only this. It's just a short list because we have roots, stems, fibers, leaves, flowers, and etc. There are many different types of macroremains which can be recovered from archaeological context. And the amount, at least 40 liters of sediment should be taken from each layer. It's my opinion. Maybe some people will be say, no, we don't agree with you. And one sample may be range between 5 to I don't know, to 100 to 200 liters. It depends how this context is important. Why 40 liters? It doesn't mean that it should be less, more. It shouldn't be more. It will be better if you will have more from each layer. If you are taking samples from the site, which has many, stratigraphically, you can see many cultural layers, it would be better to take more. Yes, I'm the person who prefer to take as much as possible. And what I would like to point out that if you find samples in water, like in wet environment, please don't take them out and put in a box, I don't know, or you shouldn't make them dry, because later you will find them broken, nothing will be to study. It should be preserved in a wet environment as well as it was in the in archaeological site. And the purpose to study seeds, fruits, macroremains, first of all, it allows us to identify cultivated plants, also to understand the origin and development of agriculture and pastoral practices, also for diet reconstruction. You can understand which plants were consumed by people or by animal. And also it's very important to study not only cultivated plants. You should pay attention to the very tiny seeds, which are even less than 0.5 millimeter, at least 0.1 millimeter, because they also give you information about ecology, about environment of the site in the past. And you can compare with ecology in the present day, because when you arrive to the archaeological site, first of all, you should describe the ecology. You should describe which plants are there, and then you compare already with your data. And macroremains anthropology. I'm not a specialist in anthropology. I hope that I will study anthropology very soon. And as you know, we I remember when I was just freshman student in my bachelor, I already took samples of charcoal because it's not so complicated to take very often. They're quite big and we're taking them for radiocarbon dating. But you can take recover wood, like burnt wood, not only for the radiocarbon dating, but it could be used for other purposes, for dendrochronology also, or for ecology and environment reconstruction, because you can identify wood species and or genus at least, and it also will be very helpful for you. Therefore, I wrote that not only an archaeobotanist, but also an archaeologist is able to recover the sample. And the amount, it's again, this information was given by our professors, and it was up to a minimum of 5 millimeters if you would like to make anthropological analysis, and up to a minimum of a few centimeters of the wood if it was selected for dendrochronology. Yes, and please avoid fragmentation if it was again found in a wet environment, keep it in a wet environment. And for dendrochronology, picking and fast shipping is important. And how actually the process is going in archaeobotany. It's sometimes it sounds so complicated. People think that if you're an archaeobotanist, probably you're like super like smart. No, I'm not. For example, I'm not a very smart person, and it's easy to do. It's kind of actually you should follow 3 steps. First is recovery. You should just come, first of all, you should visit this site before to read some papers about this archaeological site in order to understand what is your research question. It's very important to have a research question before you go to the field. Then you have to decide how many samples you need in order to answer, to get answers to your questions, and how many samples I don't know, could be stored for the future analysis. And the most important is which context have archaeologically significance to your project. And then you take samples, you can apply dry sieving, wet sieving, it depends from the environment. Do a flotation, there are different types of flotation, back flotation, and hand flotation, many different types of flotation. And then identification. Identification is a bit more complicated than recovery, because you should spend a lot of time to sort your samples and then identify them. It also takes not too much time, and then count them and create the data. But the most complicated part for me is interpretation, because you spend a lot of time trying to interpret data. You should go back to many archaeological reports, and you should just sometimes check your research question again and again. But it's how the process is going on. And what about the states of preservation? Macrobotanical remains decompose as a result of biological, chemical, or geochemical weathering process. Soil-based bacteria, different fungi, insects, or other invertebrates are primary agents that break down the anatomical structure of this deposited plant. In general, it's in all books it's written that in the warmer or wetter or more alkaline, I mean, whereas the pH is very high, and that have a high soil carbon and nutrient content will result in faster decomposition. Woody parts, like some wood remains, those high in lignin will take longer break, for example, than leaves. Therefore, it's possible to find wood, but leaves, no, if they're not carbonized or mineralized, or if they're not desiccated, it's not possible to find. And plants will preserve best in the environment that is lacking, that are lacking moisture, oxygen, have a high freezing temperature, and have a acidic and nutrient poor substates. And we have a few states of preservation. As you can see on the slide, it's carbonization, mineralization, waterlogged materials, desiccation, and freezing. In many papers and books, people, they do not separate waterlogged and waterlogging and freezing. They consider it as a together, but for me, it's a bit different. And let's start with the carbonization. Carbonization is the most common way of preservation. You can find in a wide variety of environments, because once it's burned, it will be preserved, but not always, because it could be broken down in alkaline context as well. And also it's very vulnerable to mechanical damage. And one of the, it's not a disadvantage, I don't know how to say, is that just only a small portion of a original assemblage preserved, because you can find carbonized plant remains only in ovens, in hearths, in the fireplaces, which came to the to this context accidentally during cooking, I don't know, firing, or sometimes you can find a big assemblage, a huge assemblage, if the a fire, the village was burned or the house was burned, and you can find storage places filled up with grains. It will be amazing. And also what is important is that carbonization actually shrinks specimens. And there are very different carbonization conditions. People we are trying to compare seed sizes, so to understand, for example, why wheat grains in this region is smaller than in this region. Yes, it's interesting to understand, but we shouldn't forget about taphonomic process. We shouldn't forget about the conditions, when they were carbonized, about the soil, about the temperature, about the if it was in a wet place or not. And DNA, lipids, and other molecules are destroyed in carbonized plant remains in charred remains. And the next one is mineralization, is a rare case. This occurs through the mineralization, which organic material within plant structure is gradually replaced by precipitated minerals from the surrounding area, substrate, preserving the anatomical structure of the seed or of the plant. And the process occurs frequently in a phosphate-rich context. Plants are in direct association with a corroding metal like bronze, iron, or copper. It's also one of the points. And desiccation. In other words, also people call it desiccation, mummification, dry preservation. Therefore, when you're reading archaeobotanical papers, you should not surprise. All those terms are possible and are correct. You can just accept it. And dry preservation, since almost all botanical specimens are preserved desiccated assemblage, are frequently dense and species rich. It's a big happiness to find desiccated plant because they will be like in a well state of preservation. It's easy to identify to the species level very often. And dry preservation is typically associated with desert environment, where rainfall and humidity are low. Also, it's possible to find them in the caves, like in the caves, but it depends again on different environmental conditions. For example, in ancient Egypt and in the Xinjiang region of China, like many desiccated plant remains were found. Also, despite its common association with arid environment, dry preservation also may occur in regions with a high rainfall, like in temperate regions. And if archaeological site is sufficiently protected, then it's possible to find. It's possible to find like in open air sites, for example, coastal Peru, and what I want to point out also is that water, how desiccation is actually happens, like water evaporates from the plant. And this process causes like shrinking and twisting, particularly softer, moisture-rich plant parts such as leaves, fruits, flowers, except of wood and seeds. I also wanted just to that it's possible to use for isotopic analysis. And waterlogged materials. Also, it's in some papers you can find out that people use like wet preservation, and plants also can be preserved from decomposing within an anaerobic environment. And water, of course, the chemical composition of water is also very important, and it should be also taken into consideration. But waterlogged assemblages are generally large and species rich as desiccated one. But density may be very different. The best anaerobic preservation occurs in peat bogs because of highly acidic wetlands, like with low reduction. But the problem is that these areas rarely include habitation sites. You find in the different places, but not in the habitation sites. But there are some exceptions, of course. Also, waterlogged sites in a colder climates with a minimum circulation and inflow are better preserved. Like, it's not the best preservation, but still better. And okay, it's cause degradation of molecules, but there are still some, I read some papers where they mentioned that they could extract, like DNA was recovered, have been recovered from those materials. And freezing. Freezing is a kind of also what is a part of waterlogging state of preservation, but it's a bit different for me because, yes, technically it's the sample is in a wet environment, but in this context, temperature is the main contributor. And actually it's in the ice, and we can find together with some mummies and it has an excellent preservation of biomolecules, and it's possible to apply other chemical analysis and to get more information about plants. And about techniques like which we're actually using as just a light transmitting microscope. It's used for identification. If we try to identify small macroremains, phytoliths, pollens, starch, I don't know, diatoms. And stereo microscope is mainly used for identification macroremains, but except of wood, for anthrocological analysis, again, we need a light transmitting, light transmitting microscope. And also scanning electron microscope is also sometimes used, but it's not necessary because in some labs there is no scanning electron microscope, and they still publish good papers because it's not important. It's just for some extra analysis, like analysis for extra identification. And the digital microscope. It's my favorite now because you can identify, used to identify seeds, but also you can make very good pictures of your findings of your plants, and you can make 3D models. It's good for presentations if you just make presentations in some conferences. It's good to show, and if you discuss about the morphology of the seed or nuts, fruits, it's better to use this digital microscope. Yes, and I would like to suggest you, if you just start your your career in archaeobotany, is to take pictures of all your identified seeds, and if you are publishing papers, just add them to supplementary materials, because it will be good not only for you, that your reviewers will not ask many questions about are you sure that you recovered this species in your sample. You can, yes, look at, you can find it in supplementary material, but also it will be helpful for other people who are doing archaeobotany. They can just use it as a to compare their materials with your material. Sometimes we have many atlases and manuals how to identify seeds, but it's anyway sometimes difficult to identify, and we should go through different papers and look for pictures like carbonized plants, because atlases, all those plants are carbonized, and it's a bit difficult to identify. And thank you for your attention. If you have more questions about archaeobotany, please don't hesitate to ask me. Also, you can send me emails. I can share with you some papers. I can suggest you some books, reading books, which could be very helpful and interesting for you.
Okay. Thank you, Ms. Basira, for your presentation. That's a great presentation for real. And I need to remind all the participants, if you have questions, just submit your question in the Q&A text box. And if you have any difficulties, just send your message in the chat box. And Likewise, the attendance list will be shared at the end of the presentation, so make sure you will follow all the presentation and the webinar until the end. Okay, we will continue to the next presenter, Dr. Wawan Sujarwo. Time is yours.
Okay, thank you, Niar. Good afternoon, everyone in Indonesia and the surrounding countries, and good morning in Germany and in Europe, because this morning I got WhatsApp from Mas Erwin that there are 10 countries in our webinar, so it's mean that I have to talk totally in English. This afternoon session is very struggling. You know that it's time for sleep. This is a weekend, so people take some rest, and then. But we have a webinar today. So my name is Wawan. I'm working for the Indonesian Institute of Science. I joined this institute in 2008, and at this time I serve executive director in the Ethnobiological Society of Indonesia. And okay, I share this. Have you seen my slide? No?
Yes, yes.
Okay.
Can see.
Just make it full screen.
Yes. Okay. I need to wait a minute. I think my laptop is too old, so it's a slow response. Okay. Oops. Right. Okay. Okay, have you seen this slide? I think yes.
Yes,
Yes, yes.
Okay, today I'm going to talk about plant-based food sources, archaeobotanical approach in the case of Indonesia. And my presentation is only talk about Indonesia and only talk about the history period. Yesterday we have practice, so we know each other. So this is my second presentation for the all audience, but for the committee, this is my second presentation. So I would like to show you about the map. This is our world where we are living, right? And if we are talking about the prehistoric period, we are talking about 3.3 million years ago till to the invention of writing system. It was about 5,000 years ago. So my talk is only talk about the history period. It's been that less than 5,000 years ago. And of course, I took some picture from Google, so they are not my property. So this is some pics to make a good presentation. And from the Archaeobotany, Basira already mentioned that Archaeobotany is the term in Europe, and Paleoethnobotany the term in North America. To me, it's the same, right? Because I just realized that Paleoethnobotany is the relationship between human and plants, or plant and human interaction in the past, right? I did my ethnobotanical study. I and my colleagues have published several articles in the international journal, but I just realized, because in ethnobotany we just talk to our key informants, to our respondents, and our discussion is about, if I would say, the past probably 1, 2, or 3 previous generation. But archaeoethnobotany, it talk about 100 or 1,000 years ago. And the evidence of this field of study, let's say paleoethnobotany, is plant remains, including plant parts. There are leaves, roots, stems, fruits, seeds, pollen, and so on. But if you are doing the ethnobotanical study, you only collect the information from your informant, from your key informant, and you check the plants in the study area. That's it. But the study, we can understand the relationship between plant and people and history through fossil or plant remains. But at best, we can complement with the holy books and or reliefs. It could be a best combination. It could be a best approach to do this study. And I do not know, this is the map that I took from Google. I talk about 15 years ago. Oh, sorry, 15,000 years ago, where the last glacial period ended up about 15 years ago. But at present day, most of Sundaland had been underwater, so and became Java Sea. And we suppose, I discussed with my colleagues that perhaps in this area, which is now underwater, there are many plant remains that can answer our question, research question or hypothesis, what our ancestor, the Austronesian people, have done 100 or 1,000 years ago. So it's very challenging to find the evidence or sample for the analysis, especially in the Sundaland. Because what we believe that the civilization is in the south, not in the north. Right. The south could be a good place to study of archaeobotany. And I would like to talk about the arrival of European people for the first time, starting in the 15th century or in the 16th century. And we have been colonized by the Dutch for 350 years. So I was thinking that slowly but sure, our plant-based food sources has been replaced. Even though that our ancestors, the Austronesian people, have been in contact with other civilizations long before to the coming of the European people. We believe that our ancestors have sailed to South Africa, have sailed to Madagascar. They traded kemenyan. What is kemenyan? Stearak benzoin. They trade kemenyan or stearak benzoin in Yemen, in the Middle East, long before to the coming of the European people. We have been in contact with the people in the Indian subcontinent. And we sail to the Pacific Islands, many evidence and many artifacts that our ancestors, the Austronesian people, have visited that place. And I took this picture from the relief of Borobudur Temple. There are 5 species on that relief. And only 1 species is native to the Malaysian floristic region. And the other 4 species, like Nelumbo nucifera, this is the lotus, Borasus flabelliver, Palmyra palm, Saccharum officinarum, sugarcane, and Ficus religiosa are native to Indian region, except Saccharum officinarum is native to Papua New Guinea. And I would say that about, we realize or not, right, about 70% plant-based food sources that we eat today are introduced plant species, mainly coming from South America. Cassava, tomato, chili, sweet potato, maize, avocado, cacao, vanilla. The species are native to South America floristic regions. Like salad or onion are native to Irano-Turanian floristic region in Central Asia. Even soybean. We eat tempeh or tofu almost every day. And soybean is not native to the Malaysian region. Right. Coffee, we drink coffee every day. I drink coffee 5 cups a day. But coffee is native to Africa. Also oil palm, which is very good for Indonesian economy at this moment. And coffee and oil palm were domesticated in Bogor Botanic Garden in late 18th century. So from this point, I would say that the role of archaeobotany or paleoethnobotany is essential to reconstruct our paleo diet. And of course we don't need to go further to the prehistoric period. So if we understand what is our paleo diet before to the coming of the European people, we can reconstruct this. This finding will reconstruct what our ancestors consumed hundreds or thousands years ago. And I would say that tropical archaeobotany has its own challenges. And in the next slide, I will talk about some of food sources that we still consume till today. Let's say rice, and we know that if we are talking about the origin of rice is still debatable. Many people they have their opinion, and some evidence they found the fossil in Yangtze River in Yunnan, and another scientist found that the fossil of rice was found in Ganga River in India. So, but I think we should raise another theory that rice could be native in Sundaland. And one of the fossils we can find is Sangiran. Because the Austronesian people have been cultivating and consuming rice since 2,000 years ago. And rice grain can be found in Sangiran. Sangiran is an archaeological site located in Central Java, one of the biggest one that Indonesian has. And even there, in some study, rice was invented in 3,000 or 5,000 years ago, but there is another reference that the Austronesian people have been cultivating and consuming rice since 25,000 years ago. Juawat. Juawat is one of the oldest crops or staple food that we eat till today. But I would say that this is not our main staple food. This is only an alternative choice or an emergency situation. This species is native to Eastern Asiatic floristic region, or we can say in from China. Why this? And of course, Juawat or foxtail millet, this species is only an example because there are many kinds of millet, of course, but our ancestors consumed millet in an emergency situation, right? But why this millet is not so popular? I was thinking about the taste, and people prefer to consume rice because rice tastes better than millet. And another example is breadfruit. I took this relief, of course, from Google, not my own property. And we suppose that this relief is Artocarpus altilis, which is breadfruit. This is of course, this species is another option after rice. This species is native to New Guinea, like banana. Banana is native to New Guinea because we can find many kinds of banana in New Guinea. Also, some scientists say that coconut is also native to that place, but still debatable. I do not want to talk detail about banana and coconut because it's another topics. Sagu. For the people of Papua, sagu is obvious. Right? The species is native to eastern part of Indonesia, and till today, people still consume as the main staple food. But please don't substitute sagu with rice, especially for the people in eastern part of Indonesia, including Papua, because this is their main staple food. And then if you visit Papua, there are million hectares of sagu plantation. So I think it's enough to be their main staple food. And if we are talking about our ancestor, who we are, okay, this is one of the example that in 11th to 15th century, there was a big empire, namely Majapahit Empire. And for example, excavation has been doing to discover classical artifacts. This is only example, right? Because Indonesia is very huge country. If we compare to Europe, it's from London to Istanbul. And I took two example, Kutai and Majapahit. This is only example. Because I believe that there are many places, there are many plant remains in other part of Indonesia. But from the excavation to discover plant remains or classical artifacts, we can do analysis, like using ancient DNA, ancient protein, ancient starch, phytoliths, or plant stone, like Basira already mentioned, and structures of morphology and anatomy of huts and seeds. And from the result of this analysis, we can understand to reconstruct kinds of spice, medicines, foods, and agricultural system that practiced by our ancestors 100 or 1,000 years ago. Also, we can synchronize the result of this analysis with old manuscript or holy books or reliefs. That is a good combination, like I already mentioned in the beginning of my talk. And this is an example of palynology study, the pollen analysis that my colleagues told me that the result of the analysis is able until to the species level, so that we can understand what plant species our ancestors consumed in the past. Since pollen, starch, DNA may be well preserved for a long time, so we can do study about these topics. And this is some database I took from Google. There is pollen data. At this time, there are many databases of pollen and starch, so that we can compare our result of analysis, if we do research, of course, to the database. They're already available in PALDAT. Our findings can reveal whether our ancestors consumed plant species native to Malaysian region or not. If yes, it could be a good evidence that our ancestors consumed plant species from their own floristic region. So this could be a good things to do, because we already claim ourselves that Indonesia is second mega biodiversity country in the world after Brazil. So what contribution to food sources in the modern period? So I would say this evidence. I took this picture from Nature journal, published in 2017. We can see from this figure, no data from Indonesia. So this is a good chance, or this is a good opportunity for us to do research about palynology study. No data available from our country, from Indonesia. If we do research on palynology study, from this point we can raise the theory about the bioconnections. How biological aspect influence to the connection among countries in the modern period. This can answer when the connection happened and who influenced who. In the beginning, I talk about our ancestors that sailed to the South America, sailed to Africa, Madagascar, India, Middle East, and many small islands in the Pacifics, but no data about evidence that we analyze with the scientific approach, of course, right? In Europe and Northern America, even in Africa or in South America, there are many studies about palynology. And you can see in Russia, in Japan, and in China, Australia, but no data from our country, especially from this publication. And what is we can do in the future, in the near future? For example, in Java, I would say this is only an example. Of course, another place or another island in Indonesia, there are many plant remains or artifacts. And we suppose that many artifacts, including plant remains, pollen, stems, leaves, seeds, roots, and everything, evidence about plant parts, along the northern side of the island, especially in the eastern part of Java Island, from the Majapahit Empire, 12th to 15th century. This can be a future work to discover archaeobotany of classical Javanese. This is only the case of Java, and I'm sure the case of eastern part of Indonesia, in Sulawesi, in the Moluccas, and also in Papua. Probably many. And another example, probably in Sumatra and Borneo, it was Sundaland there. We already talked about this. And that's my presentation, and only 15 slides. So we can discuss after the presentation. So thank you very much for your attention. If you have any specific questions or if you want to contact me, I put my information in the last slide, my email, and also some of the research profile. If you want to check my publication, you can do that. Thank you. Dan saya kembalikan, oh sorry, I have to talk in English. And I give back to the moderator. Thank you for the attention. Niar, please unmute.
Niar.
Ah, sorry, it's unmute. Sorry. Thank you. Sorry, I need to repeat again. Thank you, Mr. Wawan Sujarwo, for your presentation. Well, that's the last presentation in today's webinar. So we will go ahead and take time for question now. Just a reminder, please be sure to type your questions into the question box in your control panel. And I need to remind you all that the point materials from our first speaker, Basira Mir Mahamad, consists of what is actually archaeobotany, archaeobotanical remains, what could be known from the research of archaeological botanical remains, example research on archaeobotany, and analytical technique that we can use to identify archaeobotanical remains. And for our second speakers, the point materials consist of research plants in Indonesia, plants remains for food in the past society, plants remains research for environment, possibility to study archaeology, botany in Indonesia, the challenges to study botanical remains in Indonesia Pacific, and the last one is future research focus on botanical remain studies. Well, it looks like we have a few questions. The first question comes from, wait a moment, I will I will check it. Okay, this first question comes from Marlia from Indonesia to our second speakers. Mr. Wawan, would you please to refer me any publication about the rice fossil from Sangiran, because it is so interesting to understand about flora fossil in Sangiran. Thank you.
So I have to directly answer or letter?
Just
Direct.
answer directly.
Okay. I would say to Bu Marlia that one of the important reference is pre-Columbian dispersals. That could be a good reference. And then if you are interested, I will send you a paper. That's it, about that. And all the study about the dispersal of plants is already discussed on that paper. So I have some articles that probably I can share to you. Yes, right.
And Mr. Wawan Sujarwo, thank you for your answer. But she continue her statement. She wants to know about how is the development of palynology research in Indonesia. This question also comes from Mrs. Marlia.
Yeah. The problem is I'm sure that we do research on palynology, but if we refer to the publication of Nature published in 2017, there are no data available from Indonesia. So what is the problem? The problem is our lack of ability to publish our research in the global level. So it's mean that our another research communities and other country, they do not know what we are doing in Indonesia. But I'm sure that we are some research officers under the Indonesian Ministry of, I don't know, probably from university, Indonesian University, from Indonesian Ministry, they are doing research and palynology study, of course. But if we look at the global database, there is no report from Indonesia. That's why that a global community, they do not know what we are doing. So this is our problem. But if Ibu, what's the name? Ibu Marlina, I do not know. Oh no, Ibu Viola. If Ibu Viola would like to know the progress of palynology research in Indonesia, perhaps there is a report. I'm sure it's only technical report or reports, and you can find in the library of in University of in Indonesian ministry, because I have a colleague working in the laboratory of geology quarter in Bandung. She and her research group did a research in palynology, but we in LIPI, so far I do not know about research on this topic, so I could not elaborate more about this development of palynology study in Indonesia. So this is our duty to do research. Start from now, in my opinion. And we can collaborate each other from between LIPI and university and Max Planck, coordinated by University of Hasanuddin, for example. Yeah.
That's all?
Yeah. I will send some reference to Ibu Marlia because she already shared her email address. So I have not this message. So yeah.
Okay.
Thank you.
Thank you, Mr. Wawan Sujarwo, for the great answer. Sorry, I need to clarify that I made a little mistake due to I mentioned that the second question comes from Mrs. Marlia, but it's actually from Mrs. Viola. Sorry. Okay, we need to continue our question. The third questions for Ms. Basira. There are two questions. The first one is, what is better, patholites or pollen analysis in archaeology and what analytical techniques we can use to identify of them? 2. And second, have you worked on prehistorical sample? Is any different way to take sample from each different sample, like from prehistorical or from cremation site? Okay, please, Mrs. Basira.
Thank you for your question. Yes, it's about difference. What is better, phytoliths or pollynol, like to study phytoliths or pollens? First of all, yes, I missed in my presentation, didn't say, it depends from the research question, what you would like to know. First of all, palynology is generally used to look at vegetation on a regional level, rather than provide a specific site information. If you would like to say what was like, you would like to make a vegetation reconstruction only around your archaeological sites, just in few meters, maybe it's probably not good, because a deposit of pollen will usually a combination of local pollens from contemporary vegetation, regional pollens brought by, I don't know, by water, by wind, by soil erosion, and residual pollens accumulate over time. Therefore, phytoliths studies compared to palynology could be better only if you would like to know the information about specific place, because phytoliths tend to be deposited in the soil where the plant decays, and this given more information about the location for its growth or use. It's just the difference. But again, I would like to it's the most important to say that it depends from the research question. If you would like to get information about the region, yes, palynology is better. If you would like to get information only about about your archaeological sites, then you can use phytoliths. And also, phytoliths is better for grass identification. Which analytical techniques? For example, for the Macroremains study, it's easier. We just sort and study samples. But for phytoliths and pollens, you should apply some chemical treatment before to study them. And as I already mentioned in my presentation, yes, you just use a microscope when you already extract pollens and phytoliths. You just need a microscope to study. And the last question, if I worked in the prehistoric sites and or not. First of all, now I'm working with a my project related to the Silk Road. It's a historical period. But for my master degree, I studied samples from Turkey, from Arslantepe, from a Bronze Age site, and they were carbonized. And to be honest, I don't see any differences or difficulties to study from prehistoric or from historic archaeological sites. They're the same, they're carbonized. And even in a prehistoric archaeological site, which I studied for my master degree, my samples were better preserved than my samples now in the historic, which comes from the historical periods. It depends from the environment. Yes, it's everything from the environment, and the period is not important at all. But it's my opinion. Maybe I'm not right, and some people will argue with me that period is also very important and it's influenced to the state of preservation. But I think that just environment, not more.
Okay, enough.
Sorry.
Okay, thank you, Ms. Basira, for your informative answer. Now we move to our second speakers. There is a question comes from, there is an name. He asked about the Archaeobotany research caught my attention from Indonesia. Many types of biodiversity were traded commodities in the past. usually not found lasting traces in tropical environments. I'm researching on the spice root. Do you have any information about spice pollen? Such an ethnobotany can contribute to archaeobotany. Thank you.
Okay, thank you for the question. 2 days ago, I tried to search on the internet to find the literature or reference about the palynology study in Indonesia. And I came to the conclusion that I didn't find nothing. Study of palynology in Indonesia, because like the figure that I saw on my slides, I took on nature. I will share the papers to all of you, and we already seen that no data from Indonesia. So, and if we are talking about the spice root, our evidence is only from the old manuscript that still preserved in the Kesultanan, or what we call it in kerajaan in the and probably in the eastern part of Indonesia, like many sacred documents that it can be an evidence to understand our ancestor, what they have done. And about the spice root, I'm sure that before to the coming of the European people, they have been traded. They have been in contact with the people in the Middle East and the Indian subcontinent. But if we are talking about the spice root, probably 3 days ago, I talked to there was a case, college, they are research group from Badan Archaeology, Research Center for Archaeology. And if we are talking about the spice is only traded to Europe, because like clove or nutmeg pepper, it make you feel warm. So that is a little bit different if we trade the kemenyan or for the aromatherapy to the Middle East. Because we have to distinguish two types of the commodity. If we are talking about the spice, like a clove, nutmeg, pepper, and then trade it to Europe, it's make your body warm. It's only that because in Europe it's very cold, especially in winter. But if you trade the commodity to the Middle East, that is, you trade the commodity which have can produce a good smell, like a kemenyan, or let's say sandalwood, right? Sorry. Sandalwood or garu. It's for the aromatherapy because the culture is different. But if the question is asking about, do you have any information about spice pollen? I would say no. I have no information. Even I have been searching for the literature or research about the pollen study in Indonesia. But Nature say, the publication in Nature say that there is a figure that there is no report from Indonesia. So that we can do. Start from now. We do research. Okay. Thank you.
Thank you, Mr. Wawan Sujarwo, for the great answer. Miss Basira, can you hear me? There is a question.
Yes, yes.
Okay. There is a question from Rai. Whether to identify plant remains that is used for treatment and expiation, can we take sample from the stomach from mummy as the object of archaeobotanical studies? Please.
Take samples from mummies.
Yes.
Yes, of course. There are many like studies where I mentioned about freezing, and mainly like when you find the purse, like a mummy, human remains. It's not even, yeah, the mummy. You can check the stomach, and you can find some remains of food. And sometimes it's possible even to check, to find plant remains. One of the, I don't remember the name of the archaeological site, but they recovered carex. Carex is a sedge's seeds in a mammoth's stomach. It means that probably they consumed the carex as a food, like around the place where they lived. But carex is today, like all those Cyperaceae seeds, they consider it that as a indicators of wet environment. It means it was maybe like wet, many lakes or some rivers were near this archaeological site in the past. Therefore, sedges were growing there, and finding those even small wild plants, they could understand and get information about the environment in the past and compare how it's different from today. It's if I answered to the, did I answer to the question?
The question is how to identify the plant remains that is used for treatment and as poison.
Pollen sample?
No. How to identify the plant remains that is used for treatment and as a poison.
Ada anggota neramu itu yang kidal. Ada tadi kah?
Tolong yang raise to the mute for participants.
I think the question is about poison and treatment.
Ms. Basira, can you hear me? No, you unmute your
Okay, something happened. It's unmuted. First of all, it's possible to identify if they were used. Just knowing if that you should know about this plant. If that plant was if that plant is poisoned or not, and it's possible to say just about the plant. You just identify and you find the poisoned plants. It's probably could be used, but there are many different variables which have to be considered also. Many other. It could be just accidentally comes to the archaeological sites, and it doesn't mean that it was exactly used for treatment or as a poison. And also the amount. If you find just 1-2 seeds in a sample, it doesn't make sense. But if you find like a lot of like seeds of this plant, which is actually poisoned, then yes, you can consider this as an interpretation. But again, we have to be very careful giving such interpretation and take into consideration other aspects.
Okay, Ms. Basira, for the great answer. To the both speakers, there is a question from Ahmad Zain. What are the challenges of botanical archaeology research in tropical and non-tropical countries? Is there any different challenges? Each different region which has different seasons. Maybe Ms. Basira can answer as the first.
In tropical?
Yes. What is the difference? What are the challenges of botanical archaeology in two different countries, either tropical or non-tropical countries?
I have never worked in a tropical countries. I don't know very well, but what I think that maybe they have a less like probability to find the well-preserved samples, like well-preserved plants. But I'm not sure because I didn't work there.
But how about in non-tropical countries? Maybe in Europe. What is the biggest challenges in archaeobotany?
In Europe, I don't know, but the main challenges maybe is to find well-preserved samples, because if they are not carbonized, again, it depends from the region in Europe. Europe is a huge. If you find them in caves, if you find them just in an open air archaeological sites, I guess it's easier probably in Europe to find if they are carbonized or if they are waterlogged. But if they are not carbonized, if you excavate archaeological site and it's from temperate, like temperate region, and you do not have any carbonized plant remains, then it's difficult to say something. But you can apply phytoliths or make a starch analysis or, I don't know, diatoms as a proxy approaches.
Okay, thank you, Ms. Basira. Now we move to Dr. Wawan Sujarwo. The same questions with Ms. Basira about what are the challenges of botanical archaeology research in tropical countries? Is there any different challenges each different region which has different seasons? Please.
Yes. Okay. Thank you, Nir. Can I share screen once again? Okay. Because I noticed that some of the audiences coming now is increasing the audience, so probably they missed my presentation. No? Right. So it's okay. Okay. You see this slide?
Yes. Please make
full
screen.
Okay. Okay. Right. I will make a full screen. Okay. Once again, I would like to say that this is the result from nature. I took this data from nature. Right. And many audience were asking about, do you find pollen, the development of what palynology study, and then spice root based on pollen. This is the result. And based on this publication, there is no publication from Indonesia at the global level. So it's mean that the author of this paper, they didn't find report from Indonesia. So that I would like to say that we should do research on palynology study, because pollen is one of the material that we can analyze. Not only pollen, of course, right? But pollen is one of the easiest things that we can do analyze. Like I would like to share this one. Yeah, of course, I know that we are doing our researcher and the archaeology research center on it in the Balai Archaeology about the excavation to discover plant remains or artifacts, right? But we need to do analysis from ancient DNA, ancient protein, ancient stock, phytoliths. And what is the material? The materials is a plant remains that we can analyze using this approach. Right? And then we can compare if we collect the sample from pollen, right? We can do a comparison with the database, because there are many databases, right, available in the global context. But we just our evidence at this moment is only the sacred document or holy books or reliefs. But yeah, I have already mentioned that we, of course, we did the research on palynology. but it's only the national level or even in the office level. They did research, they write the report, but it's only put the report on the library or even on their table, their own table. So, and nobody will know about what they have done. So, if you want to know what your works publish, you have to publish in the global level, so everybody in this world know what have you done with your research. That's
Okay.
The context is on only in this map. But if some question we're asking about the study, this is one of the approach that we can do. I'm sure that every year we do excavation, try to find a new artifact, a new evidence. But with that evidence, we should do analysis. Right. We should analyze that material so that we can know, we can understand. Because I have mentioned about the paleo diet, right? Because when the first arrival of the European people, I have mentioned that slowly but sure they cut our habit, because 70% what we consume today are not our own plant resources. They are introduced species coming from another continents, South America and then Central Asia, Indian subcontinent. But nobody know about rambutan, Nephelium lapaceum, that is native fruits from Indonesia. Salak, salaka, that is native fruit from Malaysian floristic region. So they only know mango. Mango is native to Indian subcontinent floristic region. So even that is in. So that's just.
Okay, thank you, Mr. Wawan. Since there are so many questions come from our participants in today's webinar, in the Zoom webinar chat, there is a question from Ms. Retno Purwanti to Mr. Wawan. Have you ever found pepper pollen in Indonesia? Are there any differences between the pepper ground in Indonesia and in India? Please.
Pepper.
Yes.
I have to stop this. Right. We have to specify what kind of peppers, because there are many peppers, white pepper, black pepper, and one of them are native to our region, floristic, Malaysian floristic region, and then if we are talking about from the palynology study, I have already told that I didn't find any study of pollen from Indonesia. But if we are talking about the pepper, of course, because before to the coming of the European people, our ancestors have traded many commodities to Indian subcontinent. So it's mean that there is a flow, genetic flow from Indonesia to India, because if we are talking about the diversity between the two floristic regions, it's similar, right? Because what? Because it's influenced by the cultures. Before Islam coming to Nusantara, to Indonesia, it was Hindu and Buddha. And Hindu and Buddha originated from Indian subcontinent. So it's mean that there is a contact, acculturation, and then trading between our ancestors, the Austronesian people, and the people from the Middle East, the people from India, or even the people from Latin America. Right. That is. If
Okay,
yeah.
Thank you. To Miss Basira, can you hear me? There is a question from Ahmad. He asked about what we can identify from charcoal material and what we can reconstruct from the charcoal, please.
It's anthropology. First of all, as I mentioned in my presentation, from the charcoal you can, if it's quite, and you have at least 5 milligrams or millimeters, you can identify the species or genus of the wood which was burned. But if you know about the species or genus, you can get a lot of information about the environment. But again, for example, I read one report about Merv archaeological site, which is along the Silk Road in Turkmenistan. And in 1994, yes, in 1994, they published paper about anthropological analysis, and they identified a pistachio tree. Like they studied charcoal, like burnt wood, and they said that, okay, we have a pistachio tree, and they start to think, but is this like a pistachio tree local or not? And they didn't find it around the site, like in this region. Then they come up with the conclusion that probably they like people in like pistachio tree wood for burning. I don't know. There are many different interpretations could be done, to be honest. But it's one of the example how people just apply anthrocology in archaeology.
Okay, enough for your answer.
Yeah, it's.
Oh, okay. Okay, thank you, Miss Basira. And the last questions in the textbook from Sony Wibisona to the both speakers. I have an interesting case from the Natuna Island. There are 2 varieties of cloth from an old garden. Can Archaeobotany studies trace the DNA or origin of this type of cloth? Maybe Miss Basira can answer as the first speakers. Sorry.
Yes, DNA analysis. If you would like apply DNA analysis for archaeobotanical materials, it's possible, but it depends from the state of preservation. If it's carbonized, it's not possible to make a DNA analysis. If it was frozen, it's possible. But yes, today DNA is used, different genetic studies are used to understand about the origin of some plants, but maybe, I don't know, but glyphs, Indonesia is the origin of glyphs, as I think. But yes, DNA is used to study the origin, but for applying for archaeobotanical samples, I don't know. It depends from the state of preservation, if it's possible or not.
Okay, Ms. Basira, now the turn of Mr. Wawan Sujarwo, please.
Okay, thank you for the question, but I have another opinion, right? And I would say that the archaeobotany approach can answer this question. Of course, it can trace, but depend on the materials. Right? What is the plant remains that we do analysis? Right? Are they pollen, or, leaves, stems, woods, and any materials that we can analyze? But depends. We can choose the methods of analysis with the material that we want to analyze. But I would say, of course, yes. With this approach, archaeobotany approach, we can transfer, of course. But still depends on the materials that we want to analyze. And another question I found that from Bu Retno Purwanti, she She was asking that the types of plants are found in inscription found from the Sriwijaya, well, Mataram and Majapahit, of course. And at the Tulang Tua site, pollen of sago plants were found in the Talang Tua inscription. This is a good information because sago is native to the eastern part of Indonesia, and Palembang is located in the western part of Indonesia. So it's been, there is a contact from the people from the east and then people from the west, because the plants is growing in the east, but the inscription is available in the west. Oh, that's what we can do is only scientific data, because at this moment we just based on the old manuscript, inscriptions, holy books, and even reliefs. But if we are talking about the data from recent results, we don't have. So this is we can start from now. We can collaborate each other to do research on this, because this is we can discover what our ancestors have been done 100 or 1,000 years ago. Thank you.
Okay, thank you, Mr. Wawan. To all the participants, the attendance list has been sent in the Zoom webinar chat. You can check it, and please fulfill the attendance list. Okay, it looks like we have covered all of the questions. Finally, we come to end of presentation. We appreciate you being here. I represent the Department of Archaeology, Faculty of Cultural Science, Universitas Hasanuddin, and Association of Indonesian Archaeology Sulampapua. Would like to say thanks again for all the presenters for their informative and interesting talk, and to the audience for your active participation. Hopefully, the presentation will be beneficial for everybody. Thank you for your attention. Thanks again for joining us today, and we will see you next time. Good afternoon.
Thank you.
Yes.
So before we end this webinar, for all participants, please on your video, we will take a picture for all participants. Thanks.
Yes, I think I've done it, Bal. Yes, I've done. You have take picture.
Okay.
Okay, so yeah, thank you so much for all the participants and also our great speaker today. We will meet again in another webinar, maybe in October and November. We still have another international webinar. Perhaps we would like to talk about prehistorical period, as well as about ceramics and also pathology. So if you have any opportunity, perhaps in the future in October or November or December, we have a next webinar. So thank you so much. This is the free time. You can talk to each other if you want to still stay in this room. And if you would like to leave this room, you can leave now. Thank you so much.
Thank you. Terima kasih semuanya. Thank you.
Thank you, Mas Wawan. Maybe in the future we can make research together.
Sure.
Yes.
Yeah. I hope we can collaborate each other, even with Basira.
Yeah.
Yes.
And thank you so much, Basira, for your time to give.
Okay, vielen Dank, Basira.
Yes, thank you. It was
Have
nice
a good weekend.
Yes, have a good weekend too. Bye-bye.